Teresa?
Yes.
Cancer is the fourth leading cause of death in developed countries. And this is because current treatments are not only inefficient, but also highly toxic. As an example, we can think about chemotherapy that acts by eliminating cancer cells and also healthy cells. And this leads to the well-known side effects that we all are aware of. Hair loss, headache, muscle pain, just to name some. Thus, there is an urgent need to develop new treatments that are very specific for cancer cells so that we can lower the side effects. And most importantly, these side effects compromise to a high point the quality of life of the patients. And this makes even more necessary to reduce this in order to gain a better cancer treatment. In order to make a cancer treatment specific, we need to be able to spot the cancer cells in between the healthy cells. And this is something like finding Waldo. We need to know how cancer cells look, which characteristics they have, and then we will be able to spot them. For the ones that didn't spot him yet, he's here. So cancer cells also present certain characteristics, like certain molecules that are expressed on their surface, and they are not present on healthy cells. And antibodies are a type of treatment that can recognize these molecules, bind to them very tightly, and recruit our immune cells to lead to cancer cell elimination. However, we haven't cured cancer yet, right? Cancer cells are super smart, and they can upregulate on the surface other molecules that are also present in healthy cells. And this makes them being camouflage or makes them being masked and pretend to be healthy cells. And by that, they just confuse completely our immune system and protects them from being eliminated. So somehow the search for Waldo scenario becomes something like this, completely crazy. And the antibodies that were super useful just don't work anymore. The last breakthrough on cancer therapy, however, was the development of immune checkpoint inhibitors, which are molecules that bind on these healthy cells and block them. And by this, they exposed again cancer cells, and the normal antibodies can work again. And this has such a meaning on cancer therapy that was actually awarded the Nobel Prize for Medicine last year. However, as you can imagine, these molecules may also present a risk for our healthy cells because they can bind on the healthy cells in our body, so we have again back the side effects. In order to make a treatment that is specific and effective, we decided to merge these two different approaches into one single molecule. This single molecule would bind to cancer cells very strongly, will detect them, find them, and bind to them. And only when this happened would block the immune checkpoint or the healthy molecule, and only on cancer cells. And by this, we achieve cancer cell specificity, cancer cell elimination, and we leave healthy cells untouched, hopefully reducing then the side effects. My name is Laia, and I performed during my PhD thesis the evaluation of these molecules at the Hopfner lab in the Gene Center. And now we are currently focused on developing the proof of concept on these molecules in animal models. And of course, we have the final goal to bring them to clinics and provide cancer patients a treatment that is specific, and most of all, it's also not toxic. And by this, we can achieve a huge anti-cancer response, and at the same time, improve the quality of life of cancer patients. Thank you for listening. If you want to know more, let me know.